Hi, my name is Manasvini. I was a research intern at the Voskobonic lab down at the Hopkins Marine Station this summer, and I'm excited to share my research on neural activity modulation via electrical stimulation in the Colonial tunicate Botulus glyceri. So what exactly is Botulus and why is it a model organism for our lab? So Botulus glyceri is a colonial filter-feeding tunicate and essentially a rising star among model organisms in regenerative biology, immunology, and neuroscience. First, its transparency and relatively simple body plan allow us to directly observe physiological processes, especially nervous system activity and stem cell-driven regeneration in real time. The whole colony is made up of individual zooids, each developing synchronously through repeated budding cycles. So at any moment, a single colony contains members at potentially different developmental stages labeled A to D. Additionally, Botulus is renowned for two things: its ability to regenerate entire body systems, including the nervous system, and its fascinating allorecognition system, where colonies distinguish self from non-self, a process central to studying immune system compatibility. Its genome shares substantial homology with humans and can offer an evolutionary bridge to vertebrate biology. And so the aim for my research was to determine how electrical stimulation affects neural patterns and behavior in Botulus. We're tracking this change using siphon contractions, which are visible movements that we think are controlled by Botulus's small nervous system, sort of as a behavioral proxy for neural activity. So for the experiment, we set up two groups, controls and the treatment group, which we call PEC. Both groups began experimentation on July 9th, and each day we filmed four time windows, early morning, morning, afternoon, and night, spanning the circadian cycle with roughly 6-hour intervals. For electrical stimulation, the treatment colonies received 3 cycles of 5-minute stimulation at 20 milliamps, each followed by 15 minutes of rest. The colonies were housed together in a flow-through tank to mimic natural feeding and waste removal processes. And initially, we had used bright white LEDs for filming, but surprisingly, this stressed the animals and made their behavior less consistent. And so we switched to red LEDs, and this significantly calmed them down, leading to what I would consider more robust data. To automate our processes, we used a custom rig that controls both lighting and camera timing. And this not only saves time, but has allowed us to capture behavioral data even at inconvenient hours, which is pretty crucial for circadian rhythm analysis. So when it came to data collection and the pipeline that we used, we collected over 2 weeks of continuous data spanning two full Botulus life cycles. Each video session documents 30 minutes of colony activity with thousands of frames per video. And so you can imagine that the data sets quickly became quite massive. And so to begin, we manually counted siphon contractions frame by frame, which is quite a slow process, but essential for validating our methods. Sometimes the colonies we notice attracted small bugs, with control groups seeming to harbor more. And these microfauna can actually trigger extra siphon contractions and muddy the behavioral data. And interestingly, higher bug load may signal poor animal health, linking colony cleanliness with behavior. And so to increase efficiency and reproducibility, I developed a computational analysis pipeline. So we used FFmpeg to decimate the videos, reduce the computational load by decreasing the frame rates. And then we used Fiji to facilitate region of interest selection, marking both the buccal and the atrial siphons for contraction detection. Thresholding and edge detection macros help automate the process, and output data are input into Igor Pro for graphical representation. And so this semi-automated workflow dramatically cut down analysis time and also reduced manual bias. I'm also piloting a simple computer vision model for ROI automation, aiming just to streamline future studies. And yeah, across the life cycle, contractions and their frequency showed a clear circadian rhythm peaking at night and early morning, and then dropping through midday and afternoon. And in comparing the PEC and control colonies, we noticed that the early results show treatment colonies often displaying a greater number of contractions with statistical significance in some windows. But these differences aren't always consistent day to day and highlights the importance of extended data collection. So if we had gathered more weeks, that would be great for, a potential future study. Animal health and microfaunal load also introduced variability, and healthy colonies produced smoother, more predictable contraction patterns, while colonies with heavy bug traffic had more erratic or more excessive contractions. So, major learning throughout this project was how these subtle changes can exert outsized influence on data. Um, and it's incredibly important to have done this study because stimulation studies here not only detail how neural circuits modulate behavior, but also help uncover fundamental principles of plasticity and health state in a system with, direct relevance to stem cell and immune research. And so combining high resolution live imaging with computational behavioral quantification. Um, my research illustrated the potential challenges of using botulism as a platform for neurobiological discovery. And yeah, this work was possible thanks to my mentors. I let Vasco Boynik, my PI, my postdoc Ronnie Solomon, Stuart Thompson, Tom Rolander, and the rest of the Vasco Boynik lab team. And thank you all for listening.